Mitochondria, the powerhouse. Mitochondria are tiny organelles found in almost all living cells. They are often called the powerhouses of the cell. This is because they are responsible for producing most of the cell's energy in the form of ATP. ATP is like a tiny battery that powers all the cell's activities. Without mitochondria, our cells wouldn't have enough energy to do their jobs. Mitochondria have a double membrane. The outer membrane is smooth, but the inner membrane is folded into structures called cristae. These folds increase the surface area available for ATP production. The electron transport system, or ETS, is a crucial process that takes place in the inner mitochondrial membrane. This system is essential for cellular respiration and energy production. Think of the ETS as a cellular highway located on the inner membrane. This highway is bustling with activity, constantly working to keep our cells energized. This highway is responsible for transporting electrons from one molecule to another. These electrons are like tiny vehicles moving along a busy road. These electrons come from the food we eat, which is broken down into smaller molecules during digestion. The nutrients from our food are converted into energy-rich electrons. As electrons travel along this highway, they release energy. This released energy is vital for various cellular functions and processes. This energy is used to pump protons, which are positively charged particles, across the inner mitochondrial membrane. This proton pumping is crucial for maintaining the cell's energy balance. Think of the protons as cars traveling on the highway. They move in a coordinated manner, ensuring the smooth functioning of cellular activities. The electron transport system, or ETS, is a crucial part of cellular respiration. It is made up of several components that work together in a highly coordinated manner. First, we have electron carriers. These are like taxis that carry electrons along the highway of the electron transport chain. They play a vital role in transferring energy. They accept electrons from one molecule and then pass them on to the next carrier in the chain, ensuring a smooth flow of electrons. Examples of these electron carriers include NADH and FAD2, which are essential for the process. Next, we have complexes. These are like toll booths along the highway where electrons must pass through. These complexes are protein structures embedded in the inner mitochondrial membrane, playing a key role in the electron transport chain. As electrons pass through these complexes, energy is released, which is then used to pump protons across the membrane, creating a proton gradient. Finally, we have ATP synthase. This is like a turbine that uses the energy from the proton gradient to produce ATP, the energy currency of the cell. It's a remarkable enzyme that acts like a rotating motor, adding a phosphate group to ADP to form ATP, thus completing the process of energy production. The pumping of protons across the membrane creates a proton gradient, which is crucial for cellular energy production. This means there is a higher concentration of protons in the inner membrane space than in the mitochondrial matrix, creating a significant difference in proton levels. This gradient is like a waterfall of potential energy, ready to be harnessed for cellular activities. The protons want to move back across the membrane to the area of lower concentration, driven by the gradient. They can only do this by flowing through ATP synthase, like water flowing over a water wheel, converting potential energy into usable energy. As protons flow through ATP synthase, it spins and generates ATP from ADP and phosphate, a process vital for energy storage. This process is called oxidative phosphorylation, and it's the main way our cells produce ATP, the energy currency of the cell. The electron transport system, ETS, and oxidative phosphorylation are essential for life, powering nearly all cellular functions. They provide the energy our cells need to function, enabling everything from muscle